I am in. Good afternoon. Welcome to NYMC's Department of Public Health's National Public Health Week Speaker Series. I'm Dr. Mark Kittleson, Chair of the Department of Public Health. Our week continues to be exciting with today's talk from Dr. Nancy Stambler. We'll be introducing Dr. Stambler shortly. Our last speaker of the week is scheduled for tomorrow and will be Dr. John Allegrante from Columbia University. On Monday, we heard from Dr. Howard Kippen from Rutgers, who gave an outstanding talk on air pollution. Yesterday, we heard from Dr. Oscar Allen, a great presentation and discussion afterwards on the role of city and county health departments. First, a few items to deal with. This is being recorded and will be posted on the department's webpage within a short time. I'll be typing that webpage or that website into the chat shortly. The event is sponsored in part by the student organization STUI, Student Healthcare Executives, led by President Brian Batista. I thank Brian and all the officers for helping make this event happen. STUI is very much involved in service to the community and campus, and during this week, they are heavily involved with Feeding Westchester. So feel free to check out STUI to see how you can help with Feeding Westchester. I will be posting the STUI website in the chat as well. We are asking that you stay muted, and if you have any questions, please type it in the chat box. At the end of this presentation, we'll have about 10 to 15 minutes of Q&A. I'll be monitoring the chat to ask the questions. And we are also being assisted by Department of Educational Media with Michael Frankfurter serving as our IT specialist. Should there be any problems with the event, such as a Zoom bombing, we will end the presentation and you will receive immediately an email with a new Zoom link. Please come back at that time with that link. Thank you, Michael, for your assistance. Finally, we are trying to comply with our accrediting agency's request to maintain contact with our alumni. If you're an alum of the MPH, MS, or DrPH, we ask that you consider completing our alumni survey located in the chat. I'll be submitting those shortly as well. There's one for the MPH and MS, and there's a second one for the DrPH. Finally, I'm minded to see if Dean Ambler isn't able to make it because he's involved with the accrediting agency. But before I turn over to Dr. Hae-Young Kim, who will introduce Dr. Stambler, I just want to welcome you, Nancy. As most of you know, she teaches for our biostatistics program, some critical classes, and we are so appreciative of all of her help and assistance for this very successful MS program. So with that, being said, Dr. Kim, please introduce our speaker.
Thank you, Dr. Kittleson. It is a great pleasure for me to introduce our speaker, Dr. Nancy Stambler. Nancy Stambler is an executive director, biometrics and progenic pharmaceuticals, located in New York. She is also adjunct as assistant professor in the Department of Public Health at the New York Medical College. And then she has been teaching the biostatistics course since 2007. And then she has a MS degree in pharmacology from the Uniformed Service University of the Health Science, and also has another MS degree in Applied Mathematics and Statistics from the Stony Brook University. And then she also has a Doctor of Public Health degree in Epidemiology from the New York Medical College. She has over 30 years of experience as a biostatistician in the clinical trial and then in the drug and medical device development at the biotechnology company. She has designed and analyzed the clinical trial for many different fields, such as motor neuron disease, oncology, obesity, and rheumatology, and many other different areas. She also has published more than 50 articles for many different journals, including Neurology, JAMA, and then Journal of Urology. So today, she is going to talk about the role of biostatistician in the drug and device development from the study design and the regulatory submission. So ladies and gentlemen, please join me in welcoming Dr. Stambler.
Okay, thank you very much, Dr. Kim and Dr. Kittleson. I'm going to put up my slides and we'll get started. All right, I hope everyone can see my slides. Thank you very much for this opportunity to talk to everybody. I'd also like to remind everybody that today is World Health Day. So happy World Health Day. I know we're all participating to improve the health of ourselves as well as our communities and our planet. So today I want to pretty much talk about what I do. I mean, I think those who know me, who've been my students and work with me, know that yes, I have a day job and I'm a biostatistician in the biotechnology business. So I really want to let you know sort of what we do, but more than that, it's more of an overview. I hope I actually inspire as opposed to scare everybody, but I want to touch on some of the important things we do. So what do biostatisticians do? Not surprisingly, we have a very virtual hat rack. We have an awful lot of things we do. But primarily, it's really about the data. I remember many years ago, the American Statistical Association used to sell T-shirts. And one of them said, "In God we trust, all others bring data." And it made me laugh, but it's true. And in development, it's really about the data that support licensing of products, drugs, biological agents, medical devices. I'm currently working with some collaborators on an artificial intelligence device for computer-assisted diagnostics. So right now, even software can be considered a medical device. So it's really interesting work. So what do we do? Well, we meet with regulators because that's part of what we do. We design studies, and the biggest piece of what we do is we implement studies. With our teams, we build databases. And the most important part of building the database is to make sure that what we collect is appropriate, clean, and accurate. And yes, we perform analyses. We talk a lot about programming in our classes. So yes, we do programming. And then finally, with results, we create summaries for publications and presentations. Sometimes we make presentations like today. We contribute to all sorts of summary reports. And of course, when we put regulatory submissions to regulatory agencies, we have to respond to reviewer questions. We have to participate as part of the review. And if there's a regulatory inspection, we have to be available to talk about our share of the work in an inspection. So not everyone does everything. Some are better at design, some are better at programmers, some are better at writing. But in a small company, somebody like me may do all of the above. And seriously, we have multiple projects. That's the way the world is today, not just at a small company. But I am always grateful to my colleagues and those who help us work together to build on each other's strengths to really be successful. So what else do we do? Yeah, we teach. Yes, of course, you know that. But we also teach in addition to my students, but we teach within our work days, team members and executives. What we really want them to understand is what methods we use and why we use them and the application of those methods. And yes, when there's changes in regulatory guidance that affect how we handle data or how we create endpoints or assess endpoints within a study, this is important education that of course needs to go to our team members. And yes, of course, my students who I truly admire and love. And why do I do this? I want to help inspire them to understand what a career in statistics can be, and they can carry on the profession for us. They'll be my collaborators. So a bit of an overview of regulated settings. Primarily, I work with the United States Food and Drug Administration, but I've also worked with Health Canada and up to a small extent with some European agencies. And all of our development is really done in compliance with all of the applicable regulations and guidances. Why do we have guidances? Well, the most important thing we need to do is protect the human subjects, or in the case of animal studies, protect the animals. Why do we do this? To deliver safe and effective products. We absolutely want to minimize risks, not only in our studies, but of course, when the products are out there to help people and to provide some benefit. So just a little bit about phases of development. Traditionally, they used to be 4 phases. Phase 1 was basically safety. Phase 2 was some safety. And then if you're looking at a biologic or pharmaceutical product, to try and figure out the doses you need and maybe the dose scheduling. Then, of course, you would move to phase 3, which would then give you more confirmatory evidence of the efficacy of the product. And then phase 4 was post-marketing. Well, you always, always, always need safety. As you can see, safety is noted in all of those stages, because safety is probably even more important than efficacy. But now the world has evolved too, so your phases aren't always so clearly defined. You can have studies that are phase 1, 2, or 2, 3. And I've actually even seen studies that kind of extend across multiple phases. So basically you can say you only did 1 study, but it took 5 years. And of course you have to have some positive agreement, at least at the confirmatory level, so that we can go forward with a licensing submission. So we have lots and lots of talks with regulators, and it happens at all phases of development. Some of it begin before your first human studies. And at the end of each phase is not necessarily strictly so, but you can ask for an end of phase meeting to talk about continued development. The most important meetings are prior to putting forward a licensing submission, and of course during regulatory review. And of course, as needed, you can actually make a request to a regulatory agency if you need to discuss development plans or a particular problem that you're encountering that you really need to discuss. So I say talk early and often. It's not as if I can literally just email somebody at a regulatory agency to do so. We do have a department that we make our discussions, and then we make our plans, and then we actually do have the discussions with the regulators. So I can't pick up the phone and call somebody. However, what we do is really plan for specific reviews. And what do we discuss? Sometimes it's the particular use of the agent or a particular population or subpopulation where it makes sense. And yes, you can have talks with the statisticians at the agencies just to talk over issues or details. And that really makes it easier if you understand exactly what they're looking for. You don't have to do things multiple times. What else do we discuss? There are requirements for development. I don't have as much to do with non-clinical and manufacturing data, but I've also been asked to consult occasionally. We collect pharmacokinetic and biodistribution data. There are specific studies, and I have worked on some of those. And to me, the biggest part of what I work on is clinical data. So we have discussions to agree on study designs, and we also agree on what the endpoints to those studies should be. And sometimes it's an iterative discussion. One of my studies took almost a year to get agreement on what the efficacy endpoint should be. And of course, there are special studies that sometimes are required in development, like cardiovascular studies. But most importantly, the biostatisticians should be part of these discussions. Maybe not for all meetings all the time, but if we're talking about design or data collection, we really need to be part of that discussion so that we all agree firsthand on what we should be doing. So a bit about study design and endpoints. The big question is, what will you use? What data do you need to support your proposed product and its indication? And then, of course, how many studies? What type of evidence you need? In addition to just statistical significance, it's got to be clinically relevant. And the development plan for every product depends on the condition, the disease, the population. And we do understand what's going on with not necessarily competitive, but similar products. If somebody else has done a study design that's been successful, why not build on it? We don't always have to invent everything from the start. And we do know that the regulators compare similar products, and we'll talk a little bit more about how they use their data in a couple minutes. So study designs, we have, of course, we talk about this in our biostatistics class and study design classes. Types of studies we do. Well, comparative studies they're still usually the most frequent, but now the question is, can you still use a placebo control? And if it's not ethical to use a placebo, if you use an active control, what's the appropriate active control? Or controls? Can you use historical data? Used to be absolutely not. Now things have changed. So now you can use historical data, but this is part of the discussion you have with your team as well as the agency. And non-comparative studies are done, particularly in oncology and in rare diseases. And for diagnostic agents, the comparator is not the agent with another agent for the most point. It really is how you can define a truth standard to assess whether the diagnostic agent is in fact performing appropriately. So as part of the design, we have to assess not just the endpoint, but sometimes it's a treatment response. Sometimes it's a survival endpoint or time to event. It might be time to progression of disease. And diagnostic performance, I know there's some epidemiology lectures that talk about that, but these are important endpoints too, depending on your product. Study protocols, a lot about study protocols. They are the rules for everything. They're very detailed. We have to specify in advance, but of course, protocols can be changed. Sometimes the world changes, sometimes the development changes, and sometimes whatever we planned out in advance isn't really working. So sometimes you make amendment. So what does a protocol give you? It gives you your inclusion and exclusion criteria. It defines the population who qualifies to be in that trial. It goes through the study procedures in very large detail. And we talk about randomization if there's a randomization phase of the study. And of course, how we mask, or sometimes it's called blinding your treatments, to assure that we try to keep the integrity of the study. Of course, it talks about dosing and the assessments that need to be made. There's a lot about safety monitoring. Finally, the data we're going to collect that support the endpoints. And yes, there's a big statistical section that talks about your analysis plans. So I'll talk a little bit about the statistical data and the details in the protocol. Endpoints and analyses have to be pre-specified. Particularly your critical, your primary, your most important secondary, and maybe even some exploratory endpoints. You have to put those methods in advance. And I still remember hearing this at a statistical meeting years ago from Dr. Tony Lockenbrook, who I believe now is at the Oregon Health Sciences, but was at the FDA. And he said, you can't shoot an arrow at a wall and then draw the bullseye around it. You have to decide what your endpoint is and its success criteria, and then if you meet it or you don't meet it. So that's really an important thought. You have to have it all in advance. What data you're going to collect is critical. You have to have safety data, of course. And of course, your efficacy data, which is really going to help you define what the product does. Now, data collection instruments, are they validated? If you don't have validated instruments, they may not accept the data you collect. Many years ago, I worked in a neurology study where we had a natural history study in advance of the actual study to help us get not only the information about the disease, but to pilot and validate the instrument we were going to use. And now it's actually standard of care. So it's important not just to validate, but to find out how useful it is. So when you're collecting data, how feasible is it? If you really can't capture that data, you have to reconsider it. And don't collect what you don't need. Everybody's got pet projects of, I want to see this and I'd like to see that. And I'm all for collecting data. But if it's not important data to collect, don't bother. You have to clean all that data. And then finally, how do you actually capture that data? So of course, now in this electronic world, we no longer have people writing on forms with pens or typing them with a typewriter. Remember those things? Maybe most of you, maybe you don't. But now direct assessment is preferred wherever we can. Obviously, it can reduce quite a bit of errors. If you can get direct collections, such as central lab data, you no longer have to figure out what somebody's written on that page. And from lab to lab, their normals are different. So there's an awful lot of steps to collecting and checking those data. And now patient-reported outcomes. If you're using a validated standard health assessment, some of those are now actually available on phones and tablets. So that's now direct capture. So that's a whole lot easier than having somebody write it on a form and then having somebody else transcribe it. So some more about the statistical details we put in protocols. Why do we do this? We want to reduce bias, right? We all understand bias exists, but we're trying to reduce it wherever we can in our assessments. Obviously, one of the important things we do is randomization if we have more than one treatment. And we all know there's many, many ways to randomize now. So simple randomization and simple allocation at a fixed proportion or probability of assessment or allocation still exists. And we all know that one-to-one is our most simple, but sometimes we want more exposure to your experimental product. Stratified is terribly important. Why do you want to stratify? You want to have balance between your treatment groups. You don't want all of the men getting treatment A and all of the women getting treatment B. And adaptive randomization is definitely well in use. And that's where your allocation changes as your data change. I've seen data allocations change to remove certain treatment groups or dosing groups. And sometimes you have to be careful. Somebody on the other side really has to be keeping an eye on it, that you don't cut down, say, your placebo group so small that you can't compare it. But it's important and it's innovative and it's in current use. So we have to maintain the blind for our studies wherever we can. Not that we're hiding something quite clearly, but in order to have an appropriate, clear, unbiased assessment. So generally the patients may not know what they're getting. You try to keep the staff from knowing what they're getting. And sometimes what you really have to do is actually have different evaluators. If your product has a side effect that might tell the investigator what treatment they're getting, you have somebody who does the safety evaluation and somebody else who does the efficacy evaluations, just to try to maintain the reduction of bias best you can. And we do interim analyses and studies. Used to be you just start your study, collect your data, at the end of the day, you're done. Those days are gone. But not all studies have interim analyses. But if we do, we have to have predefined rules. And we'll talk just a little more about that in a couple seconds. So analysis plans in your study protocol, it's brief but thorough, might run 5, 6, 7 pages. However, there's always a separate statistical analysis plan, which can be quite a volume. So what goes in there? Well, obviously sample size. You have to be able to justify what you're doing. Prior data, somebody else's data, similar disease data, anything that helps us to justify what we're going to collect and how much is critical. And I kind of like the Goldilocks principle. If your study is too small, it's not ethical. It may not answer your question. Now you're wasting your time, the investigator's time, the company's money, but more important, you're wasting the participation of your subjects, of the patients in your study. You want to be able to answer that question. But if your study is too big, then you're just wasting a lot of time and money. So you have to consider, again, when we do that, if there's a lag in efficacy time or there's a lot of dropouts, that becomes part of our assessment. One more time, have to predefine all of our study endpoints, all of them, primary efficacy, secondary, exploratory, even your safety data. Then we get to the dirty part, statistical hypotheses. You've got to state your hypotheses very clearly. You've got to put your analysis methods all in there. And this is where all the detail comes. How to preserve the overall type 1 error of your study. If you have multiple endpoints, you've got to prioritize the most important. And you can't test everything without adjustment. I know you've heard me say that in some lectures, and I'm sure you've heard it from Dr. Kim and Dr. Sheet. But you also shouldn't test everything. I still cringe when I see papers that have hundreds of p-values, and it's not appropriate. So when you summarize your data, that's why we have confidence intervals. They really tell you a lot about your data. So when we write our analysis plans, we have to define all of our methods. And basically, we write a list of all the results we're going to present. So the good news is we have a lot of regulatory guidance documents that help us, not just to write an analysis plan, but to plan the analyses. So there's good regulatory guidance out there to help us. So, okay, if we're going to have an interim analysis, the actions at the end of that analysis could be continue the study as planned, but there might be a change in design. These are called adaptive designs. You could drop a treatment, you could change an endpoint, but you can't just do that because your data says so. Again, you've got to pre-specify in advance what those scenarios might be, and then if you're going to do so, how to do so. You can increase your sample size, or you might stop the study. But if you're going to stop the study, you have to have rules, not just for adaptations, but if your study has turned out to be, you expected to see some effect, and partway through the study, you see it's not going to happen. It's appropriate to stop a study for futility. Again, it's not fair to the patients, so it's okay to do so. And some studies can be stopped early for positive efficacy. If it's really working as a blockbuster, then stop. But again, what we need to do has to be disclosed to the public. But when we're writing the plans for an interim analysis and a stopping rule, we have to say what will be disclosed. You can't just have this analysis and then tell the entire study team and all the patients what's going on. If you're going to continue the trial, you have to continue to maintain the integrity of that trial. So an interim analysis is a very careful thing. And this is why we have what's called a data and safety monitoring board. Sometimes you'll hear them called a data monitoring committee or an independent data monitoring committee. And now we've actually heard some of this in the news. As you know, some of these vaccine studies, and many, many studies have these ongoing reviews. This has been going on now for decades. It's an independent review panel, includes clinicians and biostatisticians. Primarily, they look at safety monitoring, but they can look at efficacy. And if we have a planned interim analysis, they're the ones who see that. And the study statistician from the sponsor does not see that and doesn't also do the analyses. Someone else has to do those analyses, again, to maintain the integrity of your trial. It, of course, requires a charter, more pre-specified rules, what they'll see, who presents what at which sessions, when the meetings are held, and then finally, who gets disclosure to whom. So a bit about study data handling. Once we've written our protocol, gotten our safety board together, now we need to be able to collect our data. So as I said, direct capture is preferred where we can do so, but there are still data that need to be collected from the patient record or from the patient that need to be actually entered. So now it's all in electronic systems, and there's a lot of companies that do this just for a living. We have to specify every variable, what we're collecting, its attributes, and we have to be sure that those variables can actually capture data. If you're going to capture, say, the number of migraine episodes in a month, you want to be able to count them. So if you enter the number 10 and someone else enters 11, you can categorize 10 to 11. But if you just ask them to check 1 to 2, 3 to 4, 5 to 6, you can't get means, you can't get medians. So you can categorize continuous data, but you can't do it the other way. So you really have to think about the appropriate information you need to capture and how. And of course, on the other side, now that your data is being entered for you, there's a lot of checking and cleaning. So as that database is being built, we've got to program in an awful lot of what they call edit checks. It's really range checks to make sure that it's appropriate. And then another part is patient level review. So the computer does a certain amount of checking, but then the team actually looks patient by patient at specific data. Safety reviews, is something going on in this study that we need to know, whether it's done by the safety board or it's done by a safety team. And then finally, there's statistical reviews. See, again, this is where the statisticians come in. It's usually aggregate data and it's usually completely blinded with regard to treatment. What are we looking for? We're looking for data errors. We're looking for transcription errors. We're looking for incorrect units. If suddenly you've got hematology data from 2 different countries where one country's data appears to be 10 times the other, we want to make sure that they may be capturing it in different units. So we want to be able to be sure that the units are correct, or if we need to convert it, that we can make it into something that can be analyzed. And then finally, just plain mistakes. I once saw a database that had a diastolic BP of 8. Well, that's not compatible with life. So that's what a statistical check looks for. It looks for mistakes that we can find and query and clean up before we lock our databases. And of course, finally, we have documentation of all of these things we did, as well as who locked that database and when. So in the more recent years, now we have to talk about submission data standards. So our database may collect data in a certain format, but now we've got submission standards that are required. These are now mandated regulatory submission requirements. So the acronym you'd hear is called CDISC, the Clinical Data Interchange Standards Consortium. So they're a group who work with the pharmaceutical industry as well as with the regulators to help create a common standard. So the most important ones you'll hear are the one for the raw data called the Study Data Tabulation Model. You'll also hear one for analysis data called the ADAM, the Analysis Data Model. And now more recently for non-clinical data, they've started mandating that we use a similar, if you'll excuse me, similar standard called SEND, Standard for Exchange of Non-Clinical Data. And it's an awful lot like the raw data, the Study Data Tabulation Model. So data standards, very, very, very detailed documentation. Fortunately, the FDA, as well as other regulators, give you the current standards so you can meet them. So you may have to map your raw data into that format. You have to define every variable. You have to define how every derived variable is changed and collected. And finally, how it's actually calculated. And with those data, you have to give a very detailed reviewer's guide that walks through every data set, every variable, where it comes from, what it refers to, how it's calculated. And then finally, there's a very large hyperlink definition file. Why do we do this? Well, of course, it's the law. But more than that, what we wish to do is make your regulators' review easy. If your data do not meet standards, they can refuse you, and then you have to file again, and that's never good. But more than that, you want to make it easy so they don't ask you a lot of questions, right? If they can't figure out what you did and they can't verify what you did, they will ask you to do it again, or they ask you more questions that will require additional analyses on a short time frame. So your database is locked. You want to do your analyses. You have to have verified programming. Somebody makes a mistake, somebody else has to help find that mistake. Companies now call for what they love to call top-line data, because as soon as that database is locked, somebody wants your answers in a couple of days, not a few weeks. So the programmers go through it, the statisticians go through it. Usually the senior level of the team says, okay, we believe this is correct. And it goes to executive management, and then it goes out to the world. And why do we do that? We need to plan additional trials. We need to understand what's going on for development. This company needs to decide what they're going to do. But it's when those databases are locked, things get a little bit hectic. Finally, when you've done the analyses, you put them in study reports. They again describe the study procedures, the results. So you talk about the pre-specified analyses, and then you talk about what actually happened. Something happened. Was there a randomization error? Were there dosing errors? Procedures not followed. All of those things are disclosed. And of course, there may be additional analyses we do that are data-driven. All of a sudden, this is really interesting. What can we do with these data? What else do we need to do? So exploratory analyses are also included, as well as your interpretations. And yes, we work on presentations that go to scientific meetings or publications. And now you may have to work with a commercial group. Yes, there are very strict regulatory standards. I'm sure if you hear commercials on the TV of what needs to be disclosed and what's not disclosed, but what goes in print media, what goes out to doctors, right? To someone who's going to do prescribing. So they still need data. They still need summary results. And sometimes it's these presentations are not just your study results. Sometimes they're specific applications. Or if we have new methodology, of course, you want that out there for the world. And then finally, if things look good, we want to put together a regulatory licensing submission. So the standard for submission, and this is pretty much worldwide, is what's called the ECTD, the Electronic Common Technical Document. Submissions are electronic now. You are no longer putting a truckload of paper, literally going to a regulator. But now we have to write integrated analyses in addition to our study reports. So there's safety analyses and efficacy analyses. And of course, another part of the submission is manufacturing data and non-clinical data. So for integrated analyses, you have to decide what you can integrate and what you can't. Depends on your populations, your study designs, sometimes your route of administration. You can't really pull together something intravenous with something oral. But safety data usually has some that are combined, basically all exposed. But for efficacy, you really have to specifically decide how to integrate or not integrate. And this is also following regulatory guidance or discussion with your regulatory agency. But all of these integrated analyses require plans. So you have to write more analysis plans. And yes, more programming. So that's what we get paid to do. So you put together your application. What's it got? It's got all your study reports. It's got all your protocols, all of your study manuals, and then all of those data sets, your integrated data, your study data sets, and all of that very detailed documentation. But we also give them the statistical programs that use to create the combined analysis data and those that just give you your study results. Again, you want your regulators to be able to verify what you did, because if not, they're going to be asking you more questions. And of course, meetings with regulators. Before you plan your submission, you really need to talk to them and get agreement. Do we have everything we think we need? What else do they need? Do they want additional analyses? We still have time to do them, so let's get them in there. And then finally, if your product goes under review, there are specific timed review meetings. And if you have a specific designation for an orphan drug, then sometimes you have additional meetings, more discussions. And then come the reviewer questions. Sometimes they want additional analyses and they have submission deadlines. They can say, hi, we sent you this question today and we want it by next Tuesday. And it really depends on the reviewing agencies. The FDA is really pretty flexible. And if you say, we can't do that by Tuesday, but can we do it by Thursday? They'll say, sure. And the Canadians say, you have 48 hours or we'll just turn down your application. So every country is a little different. Why do they have questions? Sometimes they want to see things a different way. Sometimes they just want a clarification. And then finally, as we get towards the end of the review, if things are in fact moving along, then we get product labeling and we have to verify every number in that label. So again, that's something we work on. Every result has to in fact be verified and correct. So you get to the end of the day, your product is approved. You're really lucky. Hooray. open the champagne, that's terrific. If not, what do you need to do? More discussions? Maybe you need more trials. Maybe you need additional analyses. Maybe there's still a path forward. And then finally, you just get back to work on all those other projects. So in summary, there's a lot of pieces to product development. And to the statistician, it's really mostly about the data, but it's not just calculating a sample size and doing programming. There's so much more. It's what do we collect? How do we collect it? Is it relevant? Is it important? Is it clean? And thank goodness we work within teams, because there's a lot of operational details and so many things are best worked out together, and we have so much appreciation for each other. So I thank you for listening, and I appreciate your time as well. So I think we still have a few minutes for questions.
Yeah, this is great. Really good job. This is outstanding. I do want to mention that Dr. Stambler does have another meeting at 4 o'clock.
But I've got a few minutes.
Yeah, but we have I have 3:42, so we have 10 minutes to let's give her 5 minutes to go from meeting to meeting. So if you have a question, either post it on the chat, or if you want to raise your hand, we can get you to ask the question.
If I can find the chat. And now I should know where the chat is. Here we go.
Yeah, there's no questions yet. So
right. So far, these are some notes. Thank you.
And by the way, I did put all of the links for the various groups there, if you can look at the chat. I also want to give a shout out to our other sister departments in the school,
yes.
PT and speech language. They are doing some things, and Dr. Shikarjian gave a link to take a look at those. Quick question I have to maybe get things started. All of this is really well detailed. I'm hoping we can get the PowerPoint
Hopefully
too.
Hopefully it doesn't scare people. I'll be, Cindy already has the PowerPoints. I made a few tweaks, but I'll be glad to send it to her.
Okay.
Go ahead.
What percentage get to the point of all this work? What percentage get to the end? Are you looking at 50%, 5%, 1%, 100?
Used to be maybe 10% would get to the end. I think there's been a lot more planning, a lot more discussion. A lot of projects, if they're really not working out, they go back to the drawing board, or they just plain say, this isn't going to work. We will stop development. And of course, sometimes the company may decide, even if the product looked good from a business standpoint, it doesn't make sense. They say there's so many other competitors out there, it's not going to be worthwhile. And that's a business decision. But from a data point of view, sometimes you need a whole lot of treatments. But it used to be maybe 10%. I think maybe now, maybe it's probably more like 30 to 50%.
Oh, good.
If you're really lucky and your studies are positive, but sometimes you think everything's great and that interim analysis happens and your safety board contacts their designated person at your company and said, surprise, this is really a bomb.
Yeah.
It's awful. It's terrible. Everyone is heartbroken. But if in fact that does happen, you're now not only saving your team's time and your company's money, you're not putting your patients through that. Why would you continue to put patients into a trial if there's no hope it's going to actually benefit them?
Yeah. Pharmaceuticals spend a lot of money, and you can see just this isn't even the lab work with the with that. It's just
work. There's there's all of the manufacturing. I maybe 5% of my time, someone from manufacturing asked me to help them assess their variability between runs. Again, there are people who do that for a living. They have statisticians who just work within the manufacturing group. I work mostly within the trials group.
Good.
Someone's raised their hand with a question.
Any questions? Oh,
yeah, I have one question. Nancy, thank you very much for great presentation. Thank you. And then my question is that, you gave the presentation more detail about those submission process. So like protocol development and everything, like also data standard. But recently, FDA authorized the emergency use for the COVID vaccine, right? So the EUA, emergency use authorization, So for my question is that is there any kind of difference for the submission or review process for the EUA compared to like standard the FDA the submission for the FDA approval? Is there kind of any difference for the EUA?
You know, I really don't think so. At least for the most part, the companies that are putting together these vaccines, bless them, have large groups of people. So as they start building their database, they immediately start building the data standards so that the standard data go to the regulatory agencies as quickly as possible. Because what they will do is look, I've read these documents, not in every single word, sadly, but they will compare across different products. So the only way that they can do that is to have standard data for which the FDA statisticians or the regulatory statisticians can do so. So it has to be in a standardized format. But the actual data standards change from month to month or year to year. And those are available on the web through the FDA's website. So we always know which versions are acceptable or not, and then what's got to be changed in our processes. But for EUA, I think you still have to get the same standard data in. You just have to process it immediately.
Yeah.
Okay.
Liv Nelson, I think, has a question. Liv.
Yes, Liv.
Hello, Dr. Nancy. I just wanted to ask, this is my first introduction to biostatistics, and I am watching this presentation for my public health class called Health Behavior. Could you maybe briefly relate some of your work and research to the behavior of those involved in your trials, maybe so in keeping it unbiased?
Oh, absolutely. You know, while I work primarily in medications, there are behavioral trials. So you're right, you have to have plans in advance to keep, if you can, a standardized assessment to reduce the bias. And wherever possible, yes, you can have different assessments with different behavioral methodologies. And not everyone will get the same one. You can even randomize who gets which assessments to reduce that bias.
Good.
I
Thank you.
hope that answers it.
Yeah, Dr. Chen, and then we'll get Dr. Shi. Dr. Chen.
Okay.
Hi, Nancy.
Hi.
Thank you so much for a great presentation. It actually opened my eyes to see how the various process that we read from the media, and actually also that's why it increased the question about the recent hot topic about AZ vaccine that has actually turned out to be a poor PR right now because
Terrible,
of
yeah.
So from your standpoint of view, because as far as I know, that pharmaceutical company from the different phases, and it spent a lot of money. So, and then it seems that AZ vaccine did not pass the third phase, or so. And also, I think Heyong mentioned about the emergency use in terms of a COVID vaccine.
Sure.
So, did the standard being changed, or from your point of view, what would going to happen is that the vaccine is not going to be abandoned, or manufacture cannot be produced, or can you share your thoughts on that? Thank you so much.
Yeah, well, again, I don't have any I don't work specifically in vaccine development, but I read the same medical journals. I can read the FDA postings. The FDA advisory committee minutes, anyone can get them. You need to do a little searching on the FDA website, but you can get their documents. But in the case of AZ, they've had some real problems. As you know, they had a part of a trial where they misdosed one of the groups, and that's been disclosed. And now what they need to do is look at all of the combined data and figure out, as a function of dose, what the efficacy may be. And more recently, that manufacturing failure we all heard about last week on the news at that manufacturing plant. Now, well, run failures happen.
Yeah.
Thank you so much.
But run failures for 15 million doses is a big deal. But
That was the Johnson
some quality issues there that need some advice there.
Yeah, that was Johnson & Johnson, wasn't it?
It was somebody working for J&J, but yeah. Luckily, there's enough out there to produce, but that's a big deal.
Yeah, I'm particularly concerned because AZ vaccine has been sold into Taiwan, and then healthcare providers, the frontline workers were asked to, to get vaccinated, and then I've been telling my parents not to get it. Thank you so much.
Right, yeah, maybe there's, that if another product is available for them.
Yeah.
I don't think it's a bad product, but we do now know that there's some safety signals out there with potential blood clotting issues, which is serious.
Yeah. We only have about 3 or so minutes left
Yeah, I know
because
there's a couple
Dr.
more
Stambler does
questions.
have another meeting, but Dr. Shi and then Liv has
Yeah, Liv had
another
another question if I have a couple.
Dr.
Okay,
Shi?
first, Nancy, thank you. That's really good. You can see our Biostat students have really sent the email, ask the presentation. Yeah, and this semester we are, Dr. King and I teaching the, the clinical trial design specifically. A lot of students interested for the emergency authorized use.
Yeah.
As just you said, I say that's not like the, reduce the, standard or the simple. I say this, the vaccine that used to, they need a much more long-term follow up the safety.
Yes.
So now we at least have certain, 3 months, used to like
Right,
3 years or more year, but given the current condition, also we still not sacrifice the, the safety. We still at least have 3 months and also large sample, like 30,000. So this is the really good presentation. Students were interested. Another you mentioned about, at the beginning you said, oh, maybe too detailed. I can tell you, never be a detail. Now our students went to the interview. I always try to prepare for them. Used to like we graduate interview, just the interview statistic. I say now you have to very detailed, see their skirt, SDTM, Adam, how to
Everything.
how to do test it. Oh, so sometimes I fight with my wife. I say, when you graduate, you went to the pharmaceutical, you did not get the testers for the SARS. You did not, you get it. Oh, he said, no. Now it's, I think the FDA, the regulatory, much, complicated and standardized. So those details really good.
Yeah, there's a lot to do, but
into the class.
But for an EUA, you're just getting the data in more quickly. It's not necessarily of different quality. It just needs to be escalated. And you're right, because of the EUA, we get much shorter follow-up. We had 3 months for the EUA. Now we have some 6-month follow-up. Of course, we have no idea what the placebos could do because those who were assigned to placebo now need to be vaccinated. These rules have changed because of the emergency.
Yeah.
But it won't change all development rules, but it will give us more flexibility.
Okay.
Used to your data lock, maybe take 3 months or get the result, and now only give you like a 24 hours.
Yeah.
So that's
Very, very fast processing.
Oh, I think our last question goes back to Liv. Do you have another question, Liv?
Yes, I did. And it kind of was building on that. So I have heard from a couple different people that have like declined the vaccine because they believe it came out too quickly and that they just don't know enough about it yet. In your professional opinion, do you believe that the vaccine was produced too fast? Do you believe that all the procedural measures and trials that you mentioned in your presentation were taken before the vaccine was mass produced? Or do you think the mass population that are getting the vaccine right now are acting in some kind of way as those trials?
Well, for the most part, these are ethical companies. They follow the regulations. They want to protect people as well as get their products out there. This was accelerated development. There's shorter follow-up. I don't think anybody's really gone through skipping steps. I don't think that there has really been anything unethical. However, things do evolve. We're looking to treat billions of people. And of course, and you learn that even when you do trials of a few hundred and then your product goes out there into the market, things you couldn't have expected are going to show up. And there will continue to be ongoing assessment of safety even after products are out there. That's part of product development. There's ongoing safety
Yep.
for many years to look at emerging data
Sure.
to see what's going on. And if
The,
something appears not to be right, products can be taken off the market. We've seen it.
Well, the other thing too is that, especially with Pfizer and Moderna, there are mRNAs.
That's
That
new.
research has been going on for 20 some years.
Right, but these
So
products are
it's
new.
just that this is the first set of vaccinations that are using that, but
Right.
it's been a couple decades of work. So
Right.
this has been fascinating. Fatima just saying was the accelerated development due to large funding, as opposed in the past funding was limited. I think that probably had
For sure.
a reason for that. So
Somebody
Nancy-
giving you billions of dollars allows you to hire a lot of experts to do
Yeah.
things together.
and a lot of statisticians. And but
Yes.
anyway, I know you have another meeting at 4.
Right.
If we were physically in Skyline, we'd be presenting you a plaque, but we have to give you a virtual plaque right now.
Okay.
But Cindy does have those in her office. She's going to be mailing them out. Thank you. This was great.
Thank
I
you. hope
appreciate the
this
attendance.
inspires people to go for a career in this. And again, even if you do one piece of this as a specialist, it's intriguing work and it's great.
We're really fortunate to have you on our staff. And so thank you, Nancy.
Well, thank you,
Dr.
everyone, for
Stambler,
your time.
appreciate your help. Okay,
All right, I'll send Cindy my latest slides and they can be put up.
great. Thank you very much.
All right.
And folks,
you.
Thank you.
thanks for attending. We'll see you tomorrow for our last day. Dr. John Allegrante from Columbia will be our speaker. So
Great.
have a good day,
Thanks
everyone.
all.
Bye now.
Thank you. Thank you.